Alright, so this is going to be the pre-lab for Virida plantae lab number 1. For Virida plantae, formerly Kingdom plantae, basically what you do is you take the entire kingdom and you're going to separate it into what they call two halves, what you would call the lesser plants and the higher plants, meaning the lower and upper plants. Lesser plants would be, once Chlorophyta is moved over from Protista, that would start off with that. So you would have Kingdom Virida plantae, you'd have Phylum Chlorophyta, then you'd have Subkingdom Streptophyta, which is then every other plant on planet Earth. Under Streptophyta then, for your lower plants, you would have it starting off with things like Chaerophytans, which are the transitional form from aquatic plants going on to land. Then you go formally on land from that point on, where you're looking at everything from, say, stuff you're looking at in this lab, which would be like Hepacticophyta, those are liverworts. Anthosaurophyta, those are hornworts. Bryophyta, those are your mosses. And then that's the first half of this lab, will be those three major phyla. From there, then, you go to a group which is going to be more or less trying to go into the transitional form from the lower plants to the upper plants. As you classify plants, there's a couple key traits that you always want to look for. Number one would be, are you terrestrial or not? So are you on land or not? That's the first one. Second one is going to be, how do you reproduce? In these lower plants, pretty much what you make are spores. So either sexual or asexual spores. Seeds do not evolve until you get to higher plants. So that's one major thing you look at. Another major thing, you went over what's called it's alternation of generations. Whenever you talk about plants, they kind of throw out the whole alternation generations idea, and they use a different term called haplodiplonic life cycle. It means the same thing. Haplodiplonic, haploid, diploid. So haploid would be the gametophyte, diploid would be the sporophyte, having the full set of genetic material for that individual. So once you're in lower plants, the gametophyte's going to be the dominant phase. As you get closer and closer to the switch to where you go to what are called the more advanced plants, the gametophyte takes more and more of a backstage, sporophyte becomes more and more dominant, till eventually when you get around ferns, it's about a 50-50 split, just before ferns actually. Once you get to ferns, then it's 100% sporophyte's the dominant phase, and therefore gametophyte takes a back seat from there on, and then that becomes a big change in evolution, because sporophytes, once the sporophyte becomes dominant, the plant can pretty much become enormous. It can become huge. Along with that same time period, the way the switch works is you look for whether or not you have conductive tissues. So a vascular system. Basically, you can equate it to like the arteries and veins in your body. So xylem and phloem have to evolve. Xylem is going to take dissolved minerals and water from the roots upwards to the places where you can do photosynthesis. Phloem is then going to take sugars, which are made via photosynthesis, as well as hormones and amino acids, and move them down to the root system. So phloem takes food and hormones and aminoproteins down, while xylem is going to take water and dissolved minerals up. Okay, so it's a two-way street. Now, that doesn't evolve until, again, we start to see where the sporophyte starts to dominate. Again, once you have vascular tissue, once you have the sporophyte being dominant, you can get pretty damn big. If you think of like a giant sequoia tree, giant redwood trees in Northern California, absolutely enormous. So, therefore, those are key traits. Another key trait I obviously said too was going to be, when do you switch from spore to seed? That's an important one too. Now, the question is, when you make a seed, what kind of seed do you make? Do you make a naked seed? That's called a gymnosperm. Do you make a seed that is coated, meaning an extra layer of protection beyond the seed coat for your dispersal? If that's the case, then you would call it angiosperm. Angiosperms would be then, the angiosperm literally means coated seed, gymnosperm means naked seed. In the notes, you'll see that angiosperms, the proper name is called anthophyta. That's the proper name of them, but angiosperm is more of the common name for them. So, once you get to angiosperm, anthophyta, not only do you have vascular tissue, sporophyte's dominant, not only do you have seed production, but now you're going to have coated seeds, and then this is the last group, the biggest group, which is also going to have flowers as well as potentially bear fruit as well. When you get the plants, what you got to understand is everything that you think you know of when you go to Publix shopping for produce, fruits and vegetables, you got to throw out the window. It's real simple. If when you cut the structure open and you find seeds, that's a fruit. If you cut it open and you don't find seeds, that's a vegetative structure. A carrot, it's a storage root. A potato is basically a storage tuber. It's part of a modified stem and as well as a root too. So therefore, if they don't have seeds in those are vegetative. All right, so to your lab. If you'll notice, I skip over chlorophyta in the lab because you already drew it or saw it in Protista. You saw Spirogyra, you saw Volvox. That doesn't change. Understand that chlorophyta is going to go from unicellular immobile to unicellular mobile to going to colonial, which would be what you saw with the Volvox as well as Spirogyra, to full-blown multicellular. It's a structure called ulva. It's sea lettuce. Grows right off the shore of Florida. Now, Chaerophytans, I didn't put anything on there for you to look at for the lab. So we go now to the group, which is now the groups which are solely 100% on land. For everything we have to point on land. So first group you see there's going to be Hepacticophyta. Hepacticophyta, common name are called liverworts. When you see the word hepactica, that means liver. So with that, you're going to see Hepacticophyta. I put it in 4 ways. Okay, so with that, you're going to have the male, the female. Once we get into plants, understand, once we get to this stage, male and female becomes very, very structurally distinguishable. The male is always going to be, well, usually when you see it on slides, it's called the antheridium. So when you see antheridium, that implies male. Archegonia is female. All right, so if we do the male Hepacticophyta. Okay, so him, when you cut them in cross-section straight down, longitudinally, they kind of look like a head of broccoli almost. You have a structure coming up in the front called a thallus, nothing that I asked you to label. But then what you see then, it kind of looks like a head of broccoli. And in that head of broccoli, understand that you're going to have photosynthetic layers all around it. No, nothing deal. No, understand there is no xylem, there is no phloem, there are no seeds. What you look for, though, there's going to be these packages up here. That's going to be the male parts. Those are the spores. So that's the male. Okay? That's the sporangium. Now, for the female is why it was called liverworts. When you see it growing in your yard, the first person that discovered it or described it says, oh, well, these leaflet structures kind of look like lobes of liver. If you ever saw The Simpsons and you saw Sideshow Bob, you know, like the dreadlocks, they look like that. They look like dreadlocks is what they do, but they were called liverworts, and that's it. So if you take that structure and you cut it straight down for the female, it kind of looks like a sad mushroom. So again, you have that thallus coming up, but then it looks like a mushroom, and it looks just sad. It looks droopy, depressed, okay? Now, underneath it then, you will see these little stalks coming off and these little balls. Those are her eggs. That's how you can tell boy, girl, well, male, female, okay? So at this point, understand we can't use the word pollen. Pollen doesn't exist. He's the male counterpart, which would basically be the spermatogonia, okay? I said spores earlier. That was incorrect. That's the spermatogonia. Now, this again would be her ova. Her archegonia, her stem cells for making eggs would be found up here. And therefore, they would eventually, when they mature, drop down, almost like, think of like an ornament hanging off a tree, like a Christmas tree. Now, also then with that, so understand, this would be the gametophyte generation, gametes, okay? Now we're also going to have sporophyte generation. So for the sporophyte generation, it has the full set of DNA. It's diploid. Basically, it looks anatomically that basically you took these two structures, put them in a bag, shook them up, and dumped it. That's what it kind of looks like for sporophyte. So with this one, you're going to have kind of like this broader broccoli head looking structure. But then coming off the bottom, there's these huge sacs. And the huge sacs hanging off the bottom, those are the spores. So it kind of looks like the two merged together and made an angry child. Okay? Now, last thing you see that I want you to label or to look at for Hepacticophyta is what is called the thallus. Thallus is the stem-like structure. Now, it's going to be green, so it's photosynthetic. So, with it, basically you're going to have 2 major layers. If you want to get technical, 3 major layers. So assume then, again, this side is facing the sun, and therefore, obviously, that side should be green, because that's where you're going to put your chlorophyll, and that's where you're going to want to, produce food. So the sun's coming from this direction, okay? So what you'll have then is you'll have an outer layer. That'll be the epidermis out here, okay? Directly below that, then, the epidermis is only 1 cell thick, usually almost transparent so light can pass through. And that second layer, maybe make it a little bigger, what you'll see in it are these large balls in that second layer. That second layer, that's the photosynthetic layer. Those are chlorophyll-bearing areas. So therefore, this layer is photosynthetic. Then below that, it's going to kind of look like chicken wire, almost like looks like a network of dead cells. This layer is for water conduction. Again, it's not xylem, it's not phloem, it's just removing water from point A to point B. Well, how do you move it? Basically just by diffusion and osmosis. That's how it's going to move. So this is going to be water conducting area down here. That's the thallus. All right, now, next group you see that I have there for you is called Anthocerophyta. Antho, think antlers, horn, okay? So Anthocerophyta are called hornworts. You'll see them in your lawn, basically, if you have a dead patch of grass, and all of a sudden you get these weird, thick, look like grass blades coming out. You're like, oh, the grass is coming. No, it's not grass, it's a weed. It's a hornwort, okay? Hornworts, though, I didn't do the gametophyte, sporophyte phase for you. All I did put on there is for your sporophyte. So, therefore, the one you're looking at is called Anthoceros. Okay, it's the most common one that you would find in your yard. Anthoceros for the sporophyte, all I want you to do is look at the sporophyte generation. So basically it looks like almost like a thermometer, like this, with then an epidermis on it, okay? Down here, you would then have what you saw in fungus. It's what's called the columella. Basically, it's the area where spores would generate from. And then there would be this cylinder in the middle, and in this cylinder, it would look like uncooked popcorn kernels in that cylinder. Well, those are the spores. Then obviously you'd have the epidermis on the outside and so on and so forth. All right, next group in the lower animals is going to be Bryophyta. Of them, probably the most common ones that you could think of, because that's just moss, okay? So the slides I chose for you is a common moss called Polytrichium. Basically, it's the moss you would see growing on the side of a tree, or if you have like an air conditioning unit where the water comes out, it's in the shade, that's what you would see. Kind of looks like a green carpet. Now, when you picture moss, you got to think, all you see is all that green fuzz. That's all gametophyte. Every so often, though, in that green fuzz, you'll see this little white sprig pop up. That's the sporophyte generation. In this case now, the gametophyte's all photosynthetic, and the sporophyte is non-photosynthetic. It's real simple. If it's green, it's going to be photosynthetic. If it lacks that chlorophyll pigment, it can't be photosynthetic, which means then the sporophyte's going to rely upon the gametophyte for its nutritional needs. All right, so for Polytrichium, for moss, I'm going to do the same thing I did for Hepaticophyta. The Anthridium, the Archegonium, the male gametophyte, the female gametophyte, and then I'm going to show you the sporophyte 2 different ways from 2 different organisms. Last but not least, then I wanted to show you the quote leaf that doesn't have a true leaf at this time. To be called a leaf, a stem, and a root, you have to have xylem and phloem. You have to have conductive tissue. So right now you can call them leaf-like, stem-like, root-like. You remember the terms from fungus. Stipe is stem-like. Rhizoid is root-like. Then you also would have leaflets. All right. So for this, for the moss, the male. Now in both cases, again, I'm trying to remember the slide I showed you. I'm going to show you whenever the male and female are not fully ripe and they haven't opened up yet to give off their reproductive tissues. So they kind of look like, think kind of like a seed, where the seed coat actually comes together, but it's not a seed. It's basically you just have this, what's called integument. It's basically a protective layer on the outside that's holding something on the inside, like a pod. All right? So for the male, what you would see, there's the integument, and there's the other integument. And it's creating, a pod to protect it while it's maturing. Now, coming off of this is a layer of tissue called a pedestal. Think, like for, a chair type of thing. Now, the most important thing, though, in the middle of this, what you would see are these long finger-like projections that come up, like these long little balloons that come up on the inside. Those long little projections come up on the inside. Those are holding the spermatogonia, the male counterpart, the antheridium. So therefore, all the little dots you see in these finger-like projections, those are sperm cells. That's how you know it's a male. For the female, same basic structure. So you have the integument. You still have the pedestal. Now in this case, we're not going to have the same structure on the inside. Think of like an old-fashioned like a perfume bottle where they're like very ornate. Think about taking like a perfume bottle, heating up the glass, and pulling it to where it almost breaks. That's kind of what they look like. So what you'll see is coming up. So it looks like these long, spindly cells. How can you tell it's a female? Because right there, in each one of those, there'll be a cluster of cells. That's either, if it's immature, that would be the mother megaspore cell called the archegonium, or if it's actually formed, that would be her egg. Okay, but there is no seed. All right, so male, female. For the sporophyte generation, for Polytrichium, the common moss, if you can picture in your brain what a squash looks like or a gourd, that's kind of what they look like. Now understand, these two would be green. They're photosynthetic. For the sporophyte generation, these two are gametophyte, this one's sporophyte. Basically what you have is like this long stalk that comes up, and then it has this gourd shape on top of it. Okay, like a peanut. However, on top of it, there's a small little cap on the columella. Now, in there, then again, you would have the epidermis on the outside, nothing special, but then you'd have what look like two long little filaments that are on the inside of the peanut. Almost looks like the filaments of a light bulb, almost. Those would be the analogous to what I drew for Anthosaurophyta. So basically, these are the spore-bearing areas, and what you would see are little dots in these little filaments, and those would be your spore-bearing areas. Okay? So therefore, whenever the sporophyte is ready, think of it like a beer stein. Whenever you have a beer stein, you want to drink it, you got to pop the lid up, and then you drink. In this case, this little cap part will break off or snap up, and therefore the spores come out. All right? Now that's all for Polytrichium. Now the other one I have there for you is it's a cousin of it called sphagnum. Sphagnum moss is the kind of stuff you would buy at like a Home Depot or a Lowe's to spread around in your yard type of thing. That would be the peat moss, okay? So sphagnum, basically take this same basic idea, and it's more like, think of like a gumball machine almost, okay? So what you would see coming up would look like this. Looks very similar to what you saw in fungi for the say the Conidiophora, the Conidia in Ascomycota, or the Sporangiophora sporangia in fungus. Now what you see here though on this one is there's like this layer that looks like tire tread around the outside of it. That layer of tire tread is called an annulus, A-N-N-U-L-U-S. Okay, it's the cap holding it together. Now this right here would be the columella. And then what you would see would look like little gumballs, or again, like uncooked popcorn kernels in a hot air popcorn popper, like with a dome, and therefore those are the spores. Okay? Again, same thing, two different species. This is Polytrichium, two different genus, I should say. This is Sphagnum, this is Polytrichium. All right. Last but not least, for Polytrichium now, for moss. There's the, quote, leaf. So when you see moss, it looks, leafy. When you cut it, it looks like think of like an orange wedge, okay? So, what we're going to do is put an outer layer of epidermis, okay? All the way, all right? Now, on the bottom, what you would see is a cluster of cells down here. These would be the water-conducting cells. Again, no xylem yet, hasn't evolved yet. Then coming off of this, you would see these long little finger-like projections coming off. And they would be grainy, and those are the photosynthetic cells. So these are photosynthetic. That's your water conducting. Then you have the epidermis on the outside. So it kind of looks like almost like an orange wedge. All right? Now, that is the first half of Rota Plantae, going for the lower animals. Let me jump forward in my manual, because from there now we go to now not the high level plants, because those would be ones that bear seeds, and that's the next lab, those that are seed bearers. For the next half, we're now going from this lower level, and we're going to start to see the evolution of xylem and phloem. You start to see that the sporophyte is gaining ground and becoming more dominant, where it's almost 50-50 and then eventually takes over. All right, so for this, I'm going to draw a general pattern for you. Now, it's not going to matter from this point on what the heck I'm describing, what creature I'm describing. It's going to be the same general pattern, so please note, it's the same general pattern, okay? It doesn't matter whether or not it's a lower or higher evolved thing. It doesn't matter whether it's a root of a rose bush or whether or not it's going to be a root-like structure of a horsetail. Doesn't make a difference. So, what I'm going to do is do a cross-section of a root. Notice the pattern, notice what I'm showing you, because it's going to be the same thing repeated over and over again. The major thing that's going to be the difference is, as evolution occurs, is going to be what occurs in the smack dab middle of my picture. When it starts to evolve, the vascular tissue evolves, it always evolves in the middle of the structure I'm going to show you. But when it evolves, it's not very geometric, it's not very, doesn't look very nice. However, as evolution continues on, it starts to become more and more and more geometric, to the point where it almost looks like wagon wheels every time. Okay, but starting off, we start off ugly. You don't start off pretty. All right. So with this, then we go into the lower ones, which are now going to start to show vascular tissue, called Lycophyta. Lycophyta are given two common names. One thing they're often called are called ground pines. So it kind of looks like almost like a little miniature pine tree growing in your yard. Another thing that they're often called are called club mosses. So it looks like a moss, but it's in the shape of like a club. Understand, they are neither pines nor are they mosses. They are their own group, and therefore their distinguishing trait is they look like a club moss or they look like a ground pine, like a pine growing on the ground. Now, for Lycophyta, notice I give you a cross-section of the rhizome, a cross-section of the, quote, stem, which is the stipe, and something called the strobilus. Strobilus is Latin to mean like cone shape or dome shape. So strobilus is used a lot. So anytime you hear the word strobilus, strobili is plural, it means the shape of like a dome or a cone. Okay, so for this, the rhizome. Okay, this is the general pattern for basically everything from here on out with slight modifications. So what you would have, let's get a better pen because that one bites. I'm just going to have a general outside area, and then I'm going to have an area on the inside. Okay, it's a cross section, looks like a big donut. Now, this layer out here is going to be one cell layer thick. That's going to be the epidermis. Okay. Now, also then, we're going to have a layer of tissue right here surrounding this little circle area in the middle. That is called the endodermis. Okay. So the epidermis is on the outside. What's that there for? Basically, it's there to be a barrier between outside and inside. They tend to create small amounts of wax. Why? Because you prevent water loss. But you don't want to make so much wax, especially in a root-like system where you don't want water to come in. However, if you're above ground, like where the stipe is, or like the leaf-like areas, the fronds, yeah, you need wax so you don't lose your water as you're doing photosynthesis. You want to retain it. This area here, this big donut in the middle, this is called the cortex. The cortex is a general storage area. You can store food there, water, pigment, whatever you want. It's a storage area, okay? Think of it this way. You've all seen a carrot, yes? Take a carrot, cut straight down. Notice on the outside of the carrot, because you didn't like shave it or anything, on the outside you have that dry outer flaky area. That's the epidermis. Then when you cut it, you have all that orange area in the middle that looks like it's sweeping water. Right, that's the cortex. It's storing the food, it's storing the water. But if you look, when you cut the carrot straight down, smack dab in the middle is a little tiny circle. That's an important circle. That is called the vascular cylinder. The vascular cylinder is what's going to house the xylem and the phloem. Another name for the vascular cylinder is called the stele. S-T-E-L-E. Okay? Same thing. Now, Problem with this structure, understand, we're trying to move food and water through this pipe tube. We don't want it to leak. So oftentimes what you will see is floating in the epidermis, you would see chunks of red or orange, just like big fat chunks just floating in there. What is that? It's fat. That fat is called suberin. Understand, suberin is hydrophobic. Water won't dissolve in water, more water dissolve in it. Therefore, what's it doing? It's creating an insulatory case around the endodermis so you can move food and water without having it leak everywhere. Okay? Now, if you take this suberin strip, the endodermis, all that together, these two together are often called the Casparian strip. But understand that Casparian strip is only this outside layer of the endodermis. The whole thing is called the vascular cylinder. Now, like I said, for whether it's a root-like structure in a club moss, a horsetail, if it's a true root like in a rose bush, you see the same general pattern. The major thing is going to be what's it look like in the middle of the vascular cylinder. That's going to be how you're going to distinguish between these things. Now, like I said, for Lycophyta, for the club mosses, xylem and phloem is going to evolve, but not very geometric. It's going to be sloppy. Keep this in your brain at all times, okay? Xylem tend to be bigger cells, and they're always toward the middle of a structure. Phloem are smaller cells, and they're more toward the periphery. Not always, but this is not always, but this is very often the case. The xylem is usually stained kind of like a really, really, really dark red maroon color almost, but they're big cells stained dark red maroon. But not always, but you go by size and placement. If they're big cells toward the middle, xylem. If they're smaller cells and they're toward the periphery, they're stained everything from like light blue to like a sea green almost. If they're smaller toward the outside, that's going to be then the phloem. All right, so for this one, for the root-like structure, the rhizoid of the club moss, it almost looks like somebody put their finger in finger paint and just kind of swirled back and forth. So you have this meandering structure that goes back and forth, back and forth. Well, if you were to look in the middle of that structure, you would see large red cells all throughout that little swirl area. Well, that is going to be the xylem, X-Y-L-E-M. Well, whereas the phloem is tucked in between the little folds, so as it kind of meanders back and forth, it makes these little switchback areas here and here and here and here, all those little cracks and crevices. So the phloem, P-H-L-O-E-M, phloem is then found tucked in between. Okay? Now, that would be for the root-like structure that you would see in the club moss, okay? But it's going to be the same thing for like a horsetail, except for it can be a slight modification, with air chambers in it. Now, for the stem of the club moss, pretty much looks the same, so I'm not going to redraw it. For the strobilus, picture in your brain a pine cone and cut it straight down lengthwise. Think that shape, okay? So, you have this little, cap of your little Christmas tree looking thing, and then basically, like that, and cut it straight down. Now, each one of these levels is called a scale. So each one of the levels is called a scale. Down the middle, you would see a structure that's for support, that's integument, but the most important thing is in each one of these little scales, you would see these large circles on either side, and they're going to be filled with a bunch of little dots. That's the sporangia, okay? So therefore, whenever you look at the next plant lab number 2, again, those are going to be reproductive structures for pine trees, it would either be pollen if it's a male, or it would be the ovule if it's a female. Okay, but now there's no seeds, so everything is spore driven. Now, the next thing you need to look at is pterophodons, which are whisk ferns, called Psilotum. I have you look at the Psilotum rhizome, which is the root structure again. Basically, it looks the same as the one I just drew for you. Again, just follow the pattern and you'll see all the parts you need to. Again, but the major thing is that vascular cylinder is going to start to become more geometric, more ordered than what you saw before with that little meandering little river. Now, for horsetails, rhizome. Now, the equistem, equus is horse. So equistem, the strobilus, strobili look just like the club moss one, so I'm not going to draw that again. However, the rhizome is going to look different. The stem's going to almost look like wagon wheels almost. So, but again, it's the same general pattern, guys. What we have is that outer epidermis. No big deal. However, underneath the epidermis, what you will see are these huge, large, empty areas. Those are air chambers. What does it do? It lightens the frame of a horsetail. Because understand, horsetails are these long whip-like plant structures which can be 15, 18 feet long. Well, if they were solid, when the wind hit them, they would snap. But because they have these air chambers in them, when the air hits them, it whips back and forth like a horse's tail. That's why they're called horsetails. Now, what you will then see, so these are air chambers. In the middle, you'll see the same thing. You'll see air chambers again in a circle. So it's becoming more and more, geometric wagon wheel-like almost. So understand then, that would mean that this is the epidermis, all this is the cortex. The vascular cylinder isn't really well defined, but it's right here. The air chambers aren't part of it. Where would you see the xylem and the phloem? Real simple. I'll use green. The phloem is going to look like these, where the air chambers are, almost it's like a little shower cap on top of each one. That would be the phloem. Well, where's the xylem? Xylem, larger cells, more toward the middle. They'd be found in between, in here. In between the air chambers. That's your xylem. So notice the xylem is more toward the middle, the phloem is more toward the outside. But it makes a zigzag pattern where they juxtapose. Phloem, xylem, phloem, xylem, phloem, xylem, phloem. Make a zigzag going around. But the phloem is always toward the outside, and the xylem is always toward the inside. All right, now would cover Pterophyta. Last thing then for the lab would then be Pterophyta, the ferns proper. Now, I'm quite sure you all know what a fern looks like. Okay, so for this, I wanted you to understand when you see the fern, that whole structure is the sporophyte generation. So you have a stipe, so you have the frond, the photosynthetic area. If you were to pull it out, you would see something called a rhizome. A rhizome is not a rhizoid. I know it's confusing. Rhizoids are root-like, hair-like structures. Rhizomes are stems that grow underground. So therefore, when you have one fern, it can run its stem underground and then make another fern. Run its stem underground, make another one. So if you ever try to clear ferns out of your backyard, you pull one fern, you find out it's daisy-chained to a bunch of other ones. Basically, it's one big-ass fern is what it is. It's just kept going along underground, popping up new ones along the way. Okay. Now, if you've ever, again, seen that, all of that is sporophyte generation, okay? Even when you take the frond and flip it over, you'll notice there's these little brown, yellowish, brown, green dots found on the underside of the frond. Well, those are called sori, sorus, S-O-R-U-S, plural is sori, S-O-R-I. What are those? Those are spore-forming bodies. Well, how then does the fern release the spores? Understand, it's built really tough, so when the wind hits it, the whole frond, like a sail, will catch the air and get pulled back. However, whenever the wind stops, it'll then jerk forward. When it jerks forward, it shoots the spores away from the fern. Now, that's all sporophyte. Most of it is sporophyte. Gametophyte is about the size of a head of an eraser, and that's the first thing. So the gametophyte for the fern is called a prothallus. Okay, the prothallus looks like a beat up heart. Imagine you're in kindergarten and you have to make your first Valentine's. Okay, your scissor skills aren't the best, so what you do is you do the best you can and you make kind of this anemic, beat up heart looking thing. Okay, that's what it looks like. All right, now with this, you got to be careful where position is. Where the heart comes down to a crook in the divot in the valley, what you'll see are large cells right here. Only there. That's the female. The female is called, again, archegonia. Okay? So where's the male? The male is found further down in the body of the heart-like structure, and you'll see kind of like these small little dot freckles further down. That's the male, and that is the antheridium. Last thing, what you'll see coming off the bottom of the prothallus look like whiskers, and those are the rhizoids, root-like structures. But again, all this is about the size of an eraser head. Last thing I have you draw are the sporangia for the, sorry, not draw, but to look and label, they're going to be the sporangia for this. So, this is the gametophyte generation. Tiny. The sporophyte generation is what you think of when you think of a fern. The whole thing, that's all sporophyte. So at this point, from ferns, sporophyte now dominates and you never look back. Xylem and phloem are very highly organized, which means these things can become enormous from this point on, meaning ferns in any plant from this point on. All right, so last thing you see is going to be for the sporangium. So what you get almost looks like, again, a little hook like this. And then what you see again, it looks like tire tread going around. Okay? That is, excuse me, the annulus. It protects it. What you would see then in the middle look like little grains of popcorn again in the middle, and those are the spores. Simple as that. Now again, whenever you see this, understand when you look at the bottom, when you look underneath the frond of a fern, and you see those little piles that look like little brown dots, that's 10, 20, 30 of these in a pile, and each one is making anywhere from a dozen to 2 dozen spores. So they're quite prolific in their reproductive capacity. All right, so that is Viridiplantae lab number 1. Viridiplantae lab number 2 picks up then with seeded plants, meaning things like cone-bearing plants, conifers, going all the way up through flowering plants. All right, see you then.